Okay, so for my project, I chose VEGFA. Um, during this presentation, we're just going to go over what VEGFA is, the main role in the body, the organisms that have it, where is it located, gene size, how does it work, and the signal pathway. And with that, we're going to look at mitosis and cell division regulation. And we're only going to look at one allele because that's the one that was most well researched and had a clear phenotype for that specific allele. So moving to the first bit, VEGFA is under the family of growth factor genes called vascular endothelial growth factors. VEGFA is responsible for vasculogenesis and angiogenesis. These are the formation of blood vessels, or through new or already existing. And we should also keep in mind that growth factors are signaling proteins that stimulate receptors for an end goal of releasing a signal to tell the cell to continue to grow. So we'll come back to this thought when we have a full in-depth diagram of the signal pathway. But that's the main points. This will also be important in early embryonic development. I'm not going to have a lot of time to go over that, but just know that obviously as an embryo is developing, there's a lot of blood vessels forming and new blood vessels growing. And under this growth factor family, one in particular is responsible for placenta growth, which obviously gives nutrients to the embryo. So that's something just a part of the growth factors. Since we're talking about blood vessels and blood and the growth factor for that, we're starting in the osteoblasts or in the bones, in the bone marrow, where this gene is going to be expressed, and these growth factors will attach to the surface of endothelial cells where the specific receptor for that growth factor is located, and they'll bind there and release a signal for the cell to grow. But more on that in a second. I didn't find anything saying that this was specifically a sex-linked gene, but it is transmitted through mitotic division. In the case of epithelial cells that it's binding to and all of that. So that makes sense. Organisms that have VEGFA are as follows. We're looking at humans specifically for this one. And VEGFA is located on chromosome 6p 21.1. Background on the chromosome, of course, you have telomeres at the end, centromere in the middle, and your Q arm is your long arm. P arm is the short arm. This will be located on the P arm. And it's a part of the HLA locus, which is just a spot in the human genome that a lot of the antigens, immunosystem responses are manufactured at. So there's 3 specific spots for the HLA, which would be the 6p 21.1, 21.2, and 21.3. And you can see exactly where it's located in these 2 pictures here. Moving on the left, you can see the VEGFA protein. It's alpha folded. That's why it has an A. And it's disulfide homodimer. Homodimer just means that there's 2 existing monomers that are exactly the same. And this protein will bind as a ligand. It'll bind to either receptor 1 or receptor 2. And while there isn't a lot of research on receptor 1, People believe that it's just meant to occupy VEGFA, so there isn't actually any response from this. It's just assumed to be used to manage extra VEGFA um, uh, in itself. But for receptor 2, angiogenesis will be the result. It will tell the cell to grow. And this is the pathway that we're looking at. And VEGFA does not use receptor 3, but VEGF-C and D do. So a full pathway here is as follows. VEGFA, the ligand, will bind to the receptor 2, the VEGF receptor 2, on the surface of epithelial cells, and it will release a bunch of different proteins and kinases, and these are all different pathways that angiogenesis can result from. I was reading that kinases will increase phosphorylation and help it to attach for longer, which is more efficient for the cell, and that the if you can see PLC on the right of the screen, that is one of the phospholipase enzymes, and that's just released and it'll aid in the growth along with everything else. And you might recognize RAS here. This is a proto-oncogene. So if that is mutated on its own, it doesn't do anything. But if it's mutated, it can result or will result in unmanaged cell proliferation, and it will turn into an oncogene. So as you can see, there's just a series of pathways that can take out just different proteins, and they all lead to angiogenesis from receptor 2. And since this is carried out cell division, there's a series of checkpoints. As you can see, the G1 and G2 are the checkpoints that are most important here because they're probably going to, well, they are going to stop cell division before it even enters mitosis, but they specifically look at cell size and growth factors. So the G1 checkpoint looks at cell size and growth factors and nutrients and DNA damage. If it has any issue with any of those, it'll enter a resting state and undergo apoptosis. So this is just resting phase G1. If it passes through this and enters synthesis, it will not be stopped until the second checkpoint at G2, where it looks specifically at the cell size, which would be a good indication at this point, and DNA replication, which will also be just wrong from if it has a mutation. There's also one last checkpoint at the end of mitosis that can just stop it from dividing right before it does finish dividing for mitosis. And if that's true, then it won't divide and the cell will also go undergo apoptosis. So moving on to the allele, found conflicting sources saying that it was type 1 and then also saying that it's type 2. Either way, the resulting phenotype for the C allele of VEGFA is retinopathy. As you can see, the eyes have blood vessels in them, so you're going to have this growth factor here. And if it's in the study that I looked at, if the C allele is present in patients, there will be more likely of a chance that retinopathy will be an issue for you. Will be an issue, and their results were statistically significant as well. And just because I think I the human gene size is as listed right here on the 43 millions, and it's going to be 16,304 bases long. So that's VEGFA. sources.